Nelson, when you're starting to record.
The recording has started.
Started, okay. Good morning to everybody, and it would be good evening in Australia. It's a pleasure and an honor to have Michael Udvardi with us for our webinars today. Michael was born in Australia and spent much time working abroad, about 8 years at the Max Planck Institute of Molecular Plant Physiology, and then 15 years at the Nobel Foundation in Oklahoma, when he led the plant biology department and was also chief scientific officer there. And now he's back to Australia. And he's an expert of legume functional genomics, but regarding many aspects. Today he will be talking to us about nitrogen fixation, but I remember him also caring for tolerance to different abiotic stresses, etc. Actually, not only legumes. So thank you again, Michael, for being with us, and please go ahead.
Well, thanks, Paolo. It's nice to see you again, and greetings everyone from the University of Queensland in Australia. It's a pleasure today to talk about some of the work we've done on functional genomics of nitrogen fixation in legumes over the past years, and also to reflect a little on what might come in the future for my group and perhaps work in this area more broadly. I need to progress here. So it's customary to acknowledge the traditional owners of the land upon which we sit in Australia, and so I would like to do that now, acknowledge the traditional owners and their custodianship of the lands upon which I'm sitting here in Brisbane. I pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and I recognise their valuable contributions to Australian and global society. In today's talk, I want to cover 3 areas. I want to review the nitrogen supply problem, if you like, for agriculture, and the importance of legumes in nitrogen supply to agricultural systems. Then I want to talk about the genetics of symbiotic nitrogen fixation, review some of my group's work in this area, but also provide a more global overview of the genetics of symbiosis. And then finally, I want to think a bit about how we might go about improving symbiotic nitrogen fixation in legumes, and that perhaps forecasts the work in my group over the next few years. So to begin with, you're probably aware that nitrogen availability in the environment is often limiting to primary productivity. And on the left side of this slide, you can see the impact of adding additional nitrogen to ecosystems, whether they're terrestrial, freshwater, or marine. And these results are really meta-analysis of several hundred studies, which invariably show that adding nitrogen to these different types of ecosystems increases primary productivity in autotrophs. That's true in terrestrial, freshwater, and marine systems. Also on this slide, you can see that adding phosphorus to these systems increases productivity, and adding both nitrogen and phosphorus often has synergistic effects. So in order to optimize the use of nitrogen in agriculture, if you like, you also have to optimize other limiting factors such as phosphorus. On the right-hand side, you can see the impact of nitrogen addition in an agricultural context, and this is in the context of corn production in the US, where without added nitrogen, one can achieve between 4 and 5 tons per hectare of grain yield, but with the addition of nitrogen, you can virtually double yield. And in this panel, you can also see the added benefit of providing enough water. So to maximize yield and the return on nitrogen investment, you also need to optimize water. So, so nitrogen is limiting in agricultural systems, and recognition of that fact led to the development of an industrial process for producing nitrogen, and then industrial scale use of nitrogen in agriculture. So in my lifetime, I happened to be born in 1961. So this left panel begins the year I was born, and in that year there were about 10 million tons of fertilizer nitrogen used on the planet. Today it's well over 100 million tons. And in that time, the population of the world has more than doubled, from about 3 billion to nearly 8 billion now, I guess. So fertilizer nitrogen was really instrumental in increasing productivity in agriculture and also supporting massive population growth over the last 60 or so years. And on the right-hand side here, you can see what's happened to relative production of, for instance, cereal grains like maize, which have basically tripled in the last 60 years, and also the relative production of animals which are fed using these grains. So chickens, for instance, have increased fourfold over this period. So industrial nitrogen fertilizers truly fueled the green revolution. And of course, plant breeders played an instrumental role in that by developing better genotypes of plants, and agronomists also contributed with better management practices. But underlying everything was increased use of nitrogen fertilizers, which enabled these massive increases. As a result of the use of fertilizers, we've now doubled the nitrogen flux in the global nitrogen cycle. So you can see in the left panel the natural inputs of nitrogen through nitrogen fixation. In the terrestrial sphere and in the marine sphere, we have loss of nitrogen through denitrification. And in brown, you can see the anthropogenic inputs of nitrogen through the Haber-Bosch process, which produces fertilizer nitrogen, through the use of nitrogen-fixing legumes in production, and also through the burning of fossil fuels, which have increased oxides of nitrogen in the atmosphere, which are also ultimately falling onto agricultural and other systems. So we've doubled the flux of nitrogen in the global nitrogen cycle, and yet in many parts of the world, as you can see in the right panel, nitrogen is still limiting for crop production, for instance, in much of Africa. So although North America and Europe and Asia have sufficient nitrogen for agriculture, other parts of the world are still insufficient in terms of nitrogen. So we have this sort of dual problem of actually overuse of nitrogen in parts of the world and underuse of nitrogen in other parts of the world. And that's creating havoc, I would say, in the global ecosystems, with nitrogen, our impact on the nitrogen cycle seen as, let's say, as important as our impact on biodiversity loss and climate change. So this paper by Röckström et al. In Nature in 2009 looked at a variety of Earth systems that are essential for human well-being, if you like, and planetary well-being, and three of those systems are in a danger zone, let's say, where the activity of those systems, climate change, biodiversity loss, and the nitrogen cycle, are not considered sustainable. In fact, agriculture is a major source of nitrogen, as I mentioned already, the major source of ammonia, nitrate, and nitrous oxide pollution. And those reactive nitrogen compounds are impacting human health, ecosystem health, and in fact, climate change as well. To the cost of tens of billions of euro per year just in Europe alone. So the cost of using these fertilizers is great in terms of their, let's say, their unintended consequences. So in contrast to the use of fertilizer nitrogen, symbiotic nitrogen fixation in legumes is a more sustainable source of nitrogen for agriculture. Prior to the 20th century, this was the primary source of nitrogen in agriculture. It contributes about 50 million tons of nitrogen to agriculture each year, compared to over 100 million tons that now is provided by fertilizer nitrogen. So legume nitrogen fixation or symbiotic nitrogen fixation is now dwarfed by fertilizer nitrogen and in some respects displaced by the use of fertilizer nitrogen. But when nitrogen is fixed in legume nodules, that nitrogen is immediately incorporated into organic form by plants and used for plant growth, used for to produce plant products that we harvest, like seeds, high protein seeds. And that nitrogen ultimately, some of that nitrogen is left in the soil for subsequent crops. The form of that nitrogen is initially organic until it's turned over in the soil and then mineralized. So that nitrogen is held quite stably in the cropping system, initially in the plants, and then as it's turned over in the soil. So the loss of nitrogen from nitrogen fixed in legumes is proportionately a lot less than the nitrogen lost from fertilizer nitrogen. And you can kind of see that in this nice summary that I took from a Zhang et al. Paper published in Nature in 2015, which looked at global inputs of nitrogen into agriculture. About 50% of nitrogen fertilizer is used in cereal crop production, or in fact, nearly 60% of total nitrogen is used in cereal production. About 40% of that nitrogen that's applied to cereals is harvested with the seed that's collected from those crops. So that's a 40% nutrient use efficiency, if you like. The surplus, or the 60% that's not captured in harvested product, is actually vulnerable to loss to the environment. And this is a large part of the problem with agriculture today, is this loss of nitrogen. In contrast, legume crops like soybean are about 80% efficient in terms of nitrogen. So soybean, for instance, fixes about 20 million tons of nitrogen per year. 16 million tons of that nitrogen is captured in soybean seed in the harvested product, and therefore only 4 million tons is subject to loss in the subsequent cycling of that nitrogen in the soil. So nitrogen use efficiency in legumes is about twice as efficient as nitrogen use efficiency in cereals, and it's one of the reasons that nitrogen provided by legumes is more sustainable than nitrogen supplied via fertilizer nitrogen. So with that as a backdrop, that sort of highlights the importance of legumes in nitrogen efficiency and sustainable nitrogen use in agriculture. What I want to do for much of the rest of the talk is to talk about our knowledge and our increase in knowledge of the genetics of nitrogen fixation over the last 20 years. So about 20 years ago, we started to have access to genome sequences for legumes, and through a variety of genetic resources, we've been able to query the function and discover the functions of many genes involved in nitrogen fixation. And I guess the ultimate reason or incentive to do this work was that we hoped that through a knowledge of gene function, we could ultimately improve nitrogen fixation in legumes. And I'll talk a little bit more about that later on in the talk. Much of the work that my group has done on the genetics of nitrogen fixation was done on one of two legumes, Medicago truncatula. The other legume that we've worked a bit on is Lotus japonicus. These are two model legumes. They have similar characteristics, which were the reasons that they were chosen as models. Both Medicago and Lotus have small diploid genomes, so they were suitable for genome sequencing in the early days of genome sequencing, where cost was largely prohibitive for sequencing of many individuals of a species. They're small diploid and inbreeding, so they were largely homozygous, which meant that genome sequence that was assembled would assemble more easily because of the largely homozygous nature of that. A number of great genetic resources have been developed in these model systems, and they've enabled genetic studies and genetic discoveries, as you'll see in a moment. In addition, my group and others were able to develop gene expression atlases for Medicago and Lotus, which enabled us to identify genes involved in nodule development, and also a variety of diverse germplasm has been developed for use in genome word association studies in Medicago and Lotus and other species, and I'll talk a little bit about those resources as well later on. So one of the tools that we developed in Medicago for genetics was a transposon insertion mutant population, which took advantage of a tobacco retrotransposon that had been introduced into Medicago, and that was mobilized through tissue culture. So this retrotransposon mobilizes through transcription of an mRNA and then reverse transcription of that RNA into double-stranded DNA, which can then insert at random in the genome. So from a starting line containing 5 TNT insertions through mobilization during tissue culture, Kiran Mysore and Jianxi Wen and colleagues at the Nobel Foundation, as well as Pascal Ratet in France, were able to develop a population of 20,000 or so regenerative lines, which together contain about 1 million TNT insertions in the genome. And these insertions are at random in the genome, so there's a high probability that any gene of interest will have an insertion somewhere within this population. So we've used this transposon population for both forward genetics, where we've screened the mutants for phenotypes of interest and then used the fact that these insertions have a known sequence to identify the flanking sequences adjacent to those insertions. But we've also used the population in reverse, where we've sequenced the insertion sites to create a database of insertion sites that enable any user to screen the population for potential insertions in any gene of interest. So we've used this population in both a forward genetics mode as well as a reverse genetics mode. And as you can see from this slide, we've identified from the forward genetics mode nearly 200 symbiotic mutants that are either defective in nodulation and or nitrogen fixation. So these plants grow poorly in the absence of mineral nitrogen. This is just a collage of some of the mutants. Panel A shows the wild type nodulated plants, but we have a variety of different mutants ranging from nodulation minus mutants, such as in panel S, with many mutants that are able to nodulate but not able to fix nitrogen, and that's typically seen through this type of phenotype in panel R, where the wild type grows well under symbiotic conditions, whereas many of our mutants are unable to grow well when they have to rely on symbiosis as their sole source of nitrogen. So this population was really a great source, and I'll talk about how we've used that population in some of the coming slides. Before I get into a description of some of the mutants that we've characterized in metacarga, I just wanted to remind you of the process of nodulation and nitrogen fixation, just as a sort of a backdrop to understand the types of mutants that I'm going to describe. So that symbiosis begins actually when the plant encounters rhizobia, nitrogen-fixing bacteria in the soil. And under nitrogen-limiting conditions, plants produce compounds called flavonoids and isoflavonoids that serve as signals to the bacteria in the soil. And these signals trigger gene expression in the bacteria, including a set of genes called nodulation or nod factor genes, nod genes, that produce compounds in the bacteria that serve as signals back to the plant. And these nod factor signals trigger cell divisions in the root that ultimately lead to nodule development, as well as triggering gene expression that enables the bacteria to invade epidermal cells through a structure called an infection thread. That infection thread eventually delivers bacteria into cortical cells of the developing nodule, and those bacteria are surrounded by a plant membrane called the symbiosome membrane that leads to the formation of an organelle or a pseudo-organelle called the symbiosome, and that's the nitrogen fixing unit of the nodule. So there are two types of nodule. Metacarga produces so-called indeterminate nodules where the meristem that's triggered by nod factors is a persistent meristem. So that nodule continues to grow to form cylindrical structures or sometimes branch structures called coralloid nodules. So that's an indeterminate nodule with a persistent meristem. Other types of legumes have a more spherical structure that's formed from a meristem that's not persistent. So the meristem is active for a short period of time, maybe a week or two, and then the nodule stops growing because of a lack of persistence of the meristem. So soybean and lotus are examples of legumes that form determinate or spherical nodules. So the next set of slides, or the next few slides, I'm going to talk a little bit about some of the genes that we've discovered using resources, genetic resources in metacarga. And I'll give one example of a lotus gene that we also discovered. But I'll start with a gene that we found to be involved in rhizobial entry and accommodation. And this gene we called vaporin. It's essential for successful infection of metacarga, not only by rhizobia, but also by arbuscular mycorrhizal fungi. So this gene is required for two types of endosymbioses. Initially, we discovered this mutant, this vaporin mutant, as a mutant in a fast neutron bombardment population, actually. So it's a physical mutagen, a neutron that causes deletions. So the vaporin mutant is a putative deletion mutant, a loss of function mutant, and that mutant was unable to form normal nodules. And when we looked at the root hair cells at the infection process in the vaporin mutant, we found that unlike the wild type, which enables the rhizobia to traverse the whole of the root hair cell into the cortex through a well-defined infection structure. This is a so-called infection thread. While the wild type has a well-defined infection thread that delivered bacteria to the cortex, the vaporin mutant has a defective infection thread, and you can see that this infection thread is not coherent. The bacteria are not transported to the base of the infection thread, and many of the infections cease or terminate within the epidermal root hair cells. So in many cases, we have infected root hairs, but they don't develop into infections of the nodule, and you can see an example of that here. You get a colony forming, but that colony is unable to form an infection thread. So when we also looked at this mutant in terms of its ability to establish the symbiosis with Glomus intraradices, and this work was done in collaboration with Maria Harrison at Cornell. If you look at the infection structures in a wild type in this top left panel, you see that 14, 28, and 42 days post inoculation with the fungus, you see hyphae, vesicles, and arbuscules, and in the wild type situation, there are many more arbuscules than either vesicles or hyphae. In the vaporin mutant, the reverse is true, or the opposite is true. So although there's a lot of hyphae produced and vesicles, there are no arbuscules produced in this vaporin mutant. So there's a symbiotic defect related to infection, not only by rhizobia, but by fungal symbionts in this vaporin mutant. And we identified the gene using a hybridization approach, and based on the fact that the deletion mutant was likely affected through loss of part or all of a gene, we were able to use hybridization to find the deletion in vaporin, and ultimately we were able to find a second independent allele in our insertion mutant population that recapitulated the phenotype. And these mutants could be complemented with the full-length vaporin gene, which just restored normal nodulation and nitrogen fixation, as well as normal arbuscule development. And that work was published in the Plant Journal, I think, in 2006. So that's one example where we used the deletion mutant resource to identify a gene involved in infection by rhizobia. Another gene that we discovered, this time with the use of the insertion mutant population, was a gene that we called regulator of symbiosome development. And initially we got interested in this gene because we had expression data from the use of an Affymetrix array that we used to identify genes induced during nodule development. And one of those genes encoded a putative transcription factor of the C2H2 family, which, as I'll show, is required for normal symbiosome development. So the RSD gene, as I mentioned, is a nodule-induced gene, and we found that it wasn't expressed in roots, but by 6 and 8 days post-inoculation, we had high expression level of this gene. The gene encodes a protein with a putative C2H2 DNA binding domain, as well as an EAR repressor domain, a putative repressor domain. So this suggested to us that this gene had an important role in nodule development, given its nodule-specific expression, but also given its putative DNA binding domains and repressor domain, we speculated that this gene, in fact, might be a transcriptional repressor, and in a moment I'll show you evidence that that's the case. So we were able to isolate 2 insertion mutants in this gene, RSD gene, one in an intron and, one in the exon and one further downstream. Both of these mutants are defective in nitrogen fixation. You can see in panel C acetylene reduction activity of the wild type R108. The RSD1 allele or mutant was unable to fix nitrogen at all, whereas the RSD2 mutant fixed nitrogen, but at a much lower rate than the wild type. And in both cases, the growth of these mutants was defective compared to the wild type under symbiotic conditions. So this gene, this RSD gene, is necessary for effective nitrogen fixation. And if you look at the cross-section of wild type and mutant nodules, one can see that the bacterial population in the mutant nodules was affected, so it was much lower than the wild type, and also the normal developmental stages of nodule development were impaired in the mutant. So whereas the wild type had a clear meristem and an invasion zone and a nitrogen fixing zone, the RSD mutant was really compromised in nodule development and also unable to sustain a nitrogen fixing population of bacteria in the nodule. Bacterial or bacteroid differentiation was also impaired in the RSD mutant. So whereas in the wild type, bacteroids undergo endoreduplication and elongation, in the RSD mutant, that wasn't the case. So the bacteroids do not develop normally. And I don't really have time to go through the details of this slide, but it shows that the RSD protein acts as a repressor in an artificial yeast system. We speculated that if RSD is a repressor of transcription, that mutation in the RSD would lead to increased expression of putative target genes of RSD, and we found one putative target of RSD, a so-called VAMP, vesicle-associated membrane protein gene, which is typically repressed during nodule development, but in the mutant, in the RSD mutant, we saw that this VAMP gene remained at a high level of expression. So we speculated that the VAMP gene was at least one of the targets of RSD repression, and in a couple of ways we showed that that's indeed the case. We, for instance, in panel D, we had a portion of the promoter of the RSD gene, which we labeled radioactively, and when we mixed that promoter with the RSD protein, so the VAMP promoter with the RSD protein, we saw a retardation of that fragment in a gel electrophoresis experiment. So the promoter is slowed down when it's binding to the RSD protein, and using unlabeled probe, we were able to bind excess RSD and therefore limit the retardation. So this EMSA assay showed that the RSD transcription factor bound to the VAMP72-1A promoter. So we have a working model for what RSD may be doing. It's a transcription factor that represses VAMP72-1A gene expression during nodule development. Interestingly, the VAMP72-1A is homologous or perhaps orthologous to a similar protein in Arabidopsis, which has been implicated in plant defense. So we think that VAMP72-1A in metacargo nodules may be involved in defense, and that RSD, by blocking the activity of this protein, may interfere with plant defense during rhizobium infection of the nodule. I should say that a couple of other VAMP72-1 isoforms from encoded by two different genes have been implicated in symbiosome formation during infection of cells. So these two vesicle-associated membrane proteins are required for symbiosome production or development as the infection thread penetrates the infected cells. So these VAMPs are presumably directing vesicles that form around the rhizobia as they enter the cell. And so these are required in contrast to the amount that's suppressed by RSD. So it's possible, as I mentioned, that this 72-1A directed vesicle trafficking is involved in defense, but it's also possible that it's simply competing with resources for these vesicles that are required to encapsulate bacteria as they enter the cell. So that those are two potential hypotheses regarding the role of RSD in vesicle trafficking. All right, so the last example of a gene that I wanted to give was the role of hemoglobins in nodules. So for a long time, it was known that the most prominent plant protein in legume nodules were hemoglobins. In fact, these hemoglobins give nodules their pink color, and they were believed to be involved in nodule homeostasis in nodules to keep oxygen levels low while allowing respiration in nodules. So in the early 2000s, a new technology called RNA interference became available and enabled us to knock down the activity of 3 symbiotic hemoglobins in Lotus japonicus. So in the left-hand panel, you can see the relative transcript level of these 3 hemoglobins, leg hemoglobin 1, 2, and 3 in the wild type in black. And we used RNA interference to inhibit or to reduce the transcript level of these 3 genes. And you can see that we reduced transcript levels nearly 2 orders of magnitude. This is a log scale. So we're essentially able to remove symbiotic hemoglobins from these nodules, and you can see that in the middle panel compared to a wild type nodule, which is pink. These mutant nodules were white, so devoid of hemoglobin, and you can see that in the Western blot on the left panel. So in the wild type, we see high levels of the hemoglobin protein. In the RNAi line, essentially no hemoglobin. And that also had a knock-on effect in terms of NIFD expression. NIFD is one of the subunits of bacterial nitrogenase, and in wild-type nodules, you get high levels of NIFD. In the hemoglobin minus lines, we had no NIFD protein. So that then explains why these plants are unable to fix nitrogen. They have no NIFD, and the reason they have no NIFD is because oxygen levels in those nodules are relatively high. So that panel in the right-hand side shows the oxygen concentration at the surface, as well as the oxygen concentration down into the center of the nodule. And in the wild-type nodule, you can see oxygen levels are very low for much of the center of the nodule, whereas in the RNAi lines, oxygen levels remain high. So what we think is happening here is, in the absence of hemoglobin, oxygen levels increase in nodules, and that then inhibits the production of active nitrogenase. Interestingly, despite the higher oxygen levels in these RNAi lines, we have lower energy status. So the ATP to ADP ratio in the RNAi lines is much lower than in the wild type. So hemoglobin is necessary for maintaining low oxygen levels, but also for getting oxygen to sites of respiration in an efficient way in order to produce ATP for nitrogenase. So hemoglobins are crucial for nitrogen fixation, and that was first demonstrated in these experiments using RNAi. So over 15 or 16 years of work at the Max Planck Institute and then at the Nobel Foundation, we discovered 16 genes that had various roles in nitrogen fixation, and without which nodules either don't form or nodules form and are non-functional. So they had roles in regulation of nitrogen fixation or in transport of various compounds to the rhizobium. So that's really the tip of the iceberg. In fact, over the last 20 years, more than 200 genes have been implicated in symbiosis, not only in Medicago and lotus, but also in soybean and other legumes, but primarily in these two model systems, using a variety of genetic resources, EMS mutagens, chemical mutagens, physical mutagens like gamma rays, and also insertion mutagens, as I've described, the TNT mutants, for instance, in Medicago, and LAW insertion mutants in lotus have been important in identifying genes involved in symbiosis. More recently, CRISPR technology has been deployed to knock out genes and determine their function in both Medicago, lotus, and other legumes. So we've got a suite of genetic tools, if you like, that enable us to discover genes involved in nitrogen fixation. As I said, over 20 years now, we've got over 200 genes involved in nitrogen fixation, and we've tried to summarize that in our review here, showing genes that have been discovered in Medicago, lotus, soybean, and common bean, color-coded to show those genes that are known to be essential. Some of the genes are not essential in one species, even though they may be essential in another, which I think is a reflection of redundancy in some species where maybe more than one gene is contributing the function, that biochemical function. So as I've alluded to in the previous slides, I'm just cognizant of the time here. I've sort of gone into 45 minutes. So I'm actually not going to try to summarize the next slides, just simply to say we've tried to put those genes into context in terms of early signaling or involvement in rhizobial infection, genes involved in transcriptional regulation, genes involved in autoregulation of nodulation, which suppress nodule development once the first few nodules are produced, genes involved in hormone metabolism and signaling have been implicated in nodule development, and many genes involved in nutrient supply to nodules and nutrient supply to the nitrogen-fixing bacteria have been described over the past few years, including transporters for various metal ions that are important cofactors in nitrogenase, substrates for respiration for bacteroids, and genes involved in nitrogen assimilation. So we've learned a lot about the genetics of nitrogen fixation over the last 20 years, but we've done very little in terms of translating that knowledge to crop improvement. So that's really our challenge at this stage: is how do we move from the knowledge that we have today to trying to improve nitrogen fixation in the future through plant breeding, for instance. And I should say that this is a complex problem. As you've seen, this is a complex trait. Hundreds of genes are required for nitrogen fixation, for nodule development and nitrogen fixation. So single gene-based approaches are unlikely to really dramatically improve nitrogen fixation. So really we have to sort of take a leaf out of the page of plant breeders who are often dealing with complex traits and using breeding approaches to try to improve complex traits. So so my work now at the University of Queensland and work of collaborators is really trying to advance the breeding of nitrogen fixation, and we propose to do that by using genomic resources, but also by using genome-wide association between variation in gene sequence and variation in the trait of nitrogen fixation. So To give you a sense of the potential to improve nitrogen fixation, I just wanted to talk about a population of Medicago genotypes that was put together by Nevin Young and his collaborators that represent much of the natural variation present in the species collected from the Mediterranean basin. So nearly 300 genotypes of Medicago were collected and characterized at the genomic level. So sequence of these genotypes was produced, which enabled the identification of around 1 million single nucleotide polymorphisms in the species. So these are variations in sequence across the genome that are represented in these different genotypes. We took that population, that so-called HapMap population, and asked the question, how much variation in nitrogen fixation effectiveness do we see in a wild population, or well, it's not really a population, it's a collection of various genotypes from a wide geographic area. So what we did to determine nitrogen fixation effectiveness was to compare biomass production in each of these genotypes in the presence of mineral nitrogen versus the absence of mineral nitrogen in the presence of rhizobia. So we're comparing plant biomass under non-symbiotic optimal growth conditions with mineral nitrogen versus biomass produced during symbiosis. And what you can see in the left panel is that plants that grow bigger on mineral nitrogen tend to also grow bigger under symbiotic conditions, but then there's a great variation in their effectiveness. So for instance, if we look at plants that produce about 0.5 grams of biomass under mineral nitrogen conditions, we see a range of biomass for these genotypes under nitrogen fixing conditions, anywhere from about 0.1 to 0.5 or 0.6 grams. So there's a range of nitrogen fixation effectiveness, and in fact the right-hand side panel represents that ratio, that biomass under nitrogen fixing conditions divided by biomass under mineral nitrogen conditions. And we can see some genotypes that are 100% efficient, so they are just as effective at producing biomass under nitrogen fixing conditions as compared to mineral nitrogen. Although the majority of plants are not 100% effective, and we have effectiveness down to about 40% effective. So you can see in this collection of genotypes of Medicago that there's a wide range of nitrogen fixation effectiveness. And if we were to use these genotypes in a breeding program, one could imagine taking genotypes that have relatively high effectiveness, crossing them and trying to get progeny with even greater effectiveness. And that's really the kind of rationale for what we want to do in future, not in model species like Medicago, but in crop species. Just to finish the Medicago story, we were able to map SNPs associated with effectiveness. So with a high number of SNPs, so about 1 million single nucleotide polymorphisms, we had a high level of resolution, and we were able to identify genes that may, or variants in genes that may account for effectiveness in Medicago, and I've listed some of those here. The one example on the left is SNPs in a molybdenum transporter that appear to associate with nitrogen fixation effectiveness, and in separate work by our collaborators in Spain, this molybdate transporter was shown to be essential for nitrogen fixation. So variation perhaps in the expression level of this gene amongst natural variants in the Medicago population seem to contribute to effectiveness. So it's one of many loci that may contribute to effectiveness in nitrogen fixation. So when you talk to plant breeders, you find that that very little effort has been devoted to trying to improve nitrogen fixation effectiveness. And plant breeders will tell you, well, if they're selecting for performance in the field under nitrogen fixing conditions, the best performers will probably be the best nitrogen fixers as well. So I think plant breeders might argue that they don't need to specifically select for nitrogen fixation in order to improve nitrogen fixation in breeding programs. I would argue that that's probably not true. And in fact, the data in this slide published from a study in Canada recently looks at the nitrogen fixation effectiveness of genotypes that have been developed over the last 100 years in Canada. And what you can see in the data here is that, well, in the top section, that the number of nodules really hasn't changed in the genotypes that have been developed from around the 1910s through the 2000s. So the number of nodules has more or less been stable, although it varies with genotype. The percentage of nitrogen derived from atmosphere, in other words, the percentage of nitrogen derived from nitrogen fixation as opposed to mineral nitrogen in the soil, also hasn't changed much in these genotypes over 100 years. So even though these genotypes have been selected for increased yield over the last 100 years, the fraction of nitrogen provided by nitrogen fixation hasn't really increased. So the total nitrogen fixed has increased, but the percentage of nitrogen fixation hasn't really increased. And that's in part, I think, because breeders haven't been selecting for nitrogen fixation effectiveness. They've simply selected on yield. So I think there's scope to improve nitrogen fixation effectiveness, and I think others believe this to be true as well. And here are 3 recent publications that have started to look at look for quantitative trait loci that underpin nitrogen fixation effectiveness in soybean and in common bean. So there is natural variation out there in crop species, and therefore the potential to harness that natural variation in breeding programs. We aim to do that in mung bean, which is one of 4 pulses that are grown in Queensland. The others include chickpea, faba bean, and soybean. We've started work on a pan-genome of mung bean. So we're sequencing multiple genotypes of the mung bean species with the view of identifying variation not only in sequence across different genotypes, but also in the presence and absence of genes. So one of the, I think, discoveries that has been made in the last few years as we sequence more and more plant genomes of any one species is that individuals of a species vary in their gene content. So some individuals may have more or fewer copies of a particular gene, and that variation in gene copy number has been associated with important traits in some species. So we're using pangenomics to identify the full suite of genes available in mung bean and potentially other species as a starting point for genome-wide association studies, and eventually modeling of the association between variation in gene sequence or gene content and nitrogen fixation effectiveness. So some of the open questions, I think, in this area of nitrogen fixation are: to what extent have changes in gene regulation or gene copy number or protein function shaped nitrogen fixation effectiveness? And that's something we hope to get through genome-wide association studies in the future. What common and unique innovations have been evolved, if you like, in different legume lineages? And so we feel that phylogenomics and comparative genomics between legumes, and especially between pangenomes of different species, will inform this question about which innovations are shared between legumes and which may be unique to particular legumes. And I guess ultimately, can we use some of the knowledge of the 200 plus genes that we now know as prior knowledge in these association genomics types of studies in order to inform predictive models of function in nitrogen fixation. So just to summarize, what I hope I've shown today is that legume reference genomes and functional genomics have enabled rapid advances in our understanding of the genetics of nitrogen fixation, but translation of that knowledge to crop improvement has been slow. So I think we need to take a new approach to improving nitrogen fixation, one that embraces pangenomics, but also that looks specifically at nitrogen fixation effectiveness as a trait in breeding programs. And I think by combining, a survey of variation in gene sequence and abundance with association genomics and more traditional breeding approaches, we can take a predictive breeding approach to improving nitrogen fixation in legumes. So with that, I'd like to thank members of my former group at the Nobel Foundation for their contribution to the work I discussed today. Also, my collaborators in North America and Europe, also funding through the National Science Foundation and through the Nobel Foundation over the many years of my work there. With that, Paolo, I think that's it from me. Thank you for your attention and happy to take any questions.
Thank you, Michael. Also, if I may say, for the remarkable clarity of your talk. Myself, I have a few questions for you. I think you showed the great complexity of nitrogen fixation. So as many as a few hundred genes involved that we find out not by different methods of knocking out different genes, and then we understand that we need these genes for the function. But then when we get to the issue of plant improvement, I see that you turned into the plant and into quantitative genetic approaches, because a normal plant, let's say, has already normally all these genes needed for functioning. And so one first kind of background question is the following. To which extent legume plants are, let's say, normally self-sufficient for nitrogen got by nitrogen fixation, meaning that is really nitrogen fixation a limiting factor for plant productivity, or normally, let's say, this may not be at least often the main problem, so that there are other kind of limiting factors for legume crop production?
Yeah, that's a good question. I think for those that are trying to deploy legumes as a source of nitrogen in, for instance, in Africa, to try to improve agricultural systems there, one of the limitations that they have to overcome, or there are two major limitations, I would say. First is getting effective rhizobia onto those legumes. So especially if a legume is going into an environment where there are not suitable rhizobia, then those effective rhizobia need to be introduced with those plants. And secondly, phosphorus is often limiting in those situations. So in order to get optimal nitrogen fixation, it's been found that you also need to provide sufficient phosphorus fertilizer. So I think there are, so generally I would say that nitrogen fixation doesn't work at its full potential, either because of other limitations, and that may be limitations other than the rhizobia, such as phosphorus or water or salinity or drought. So those environmental factors limit nitrogen fixation, but but it's also true, I think, that there are intrinsic limitations in the symbiosis itself. As you can see, there's variation within a species for the ability to fix nitrogen, and therefore I think potential to increase that. So it may not really have answered your question. I'm not sure if there's a specific answer to your question, whether legumes are self-sufficient for nitrogen. I think almost by definition they are. You know, if you're not adding nitrogen to a legume crop, then they're to some extent self-sufficient. But of course, that ignores the fact that some mineral nitrogen is also present in the soil, and legumes can use both mineral nitrogen and nitrogen derived from nitrogen fixation. So yeah, generally legumes can do well and do well in nature, but I think they can do better through genetic improvement, through management, providing the right rhizobium strains, and optimizing other nutrients and other, say, environmental factors like water.
Thanks. So you did point out that improving nitrogen fixation is absolutely essential when we have new introductions of crops in which we wouldn't have, let's say, normal rhizobia. That is clear. Besides being potentially useful also in commoner situations in which we have a long-standing, a traditional crop in a given area. And also a second kind of general question, I guess it's not really easy to have an answer, is to which extent our ordinary crop improvement based on yield or prediction of yield has this kind of correlated effect to improve also nitrogen fixation?
Yeah, well, what I tried to show with the soybean example from Canada was that, you know, when we look at genotypes that have been developed over 100 years and selected based on yield, so over that period, yield has increased. Nitrogen fixation effectiveness, so that fraction of nitrogen coming from the atmosphere, from rhizobia, hasn't really increased. So although the total nitrogen fixed has increased because plants are bigger and therefore they're able to support more nitrogen fixation, that effectiveness hasn't increased. And what I also tried to show was that there is scope to improve effectiveness, to identify alleles, haplotypes perhaps, that contribute more towards nitrogen fixation than other haplotypes. And that's kind of where we're headed, I think, is trying to identify segments of chromosomes that contain alleles that are more beneficial or that contribute more to nitrogen fixation. And that may be because the gene that's associated is expressed at a higher level, or the protein is somehow more effective. And these are the sorts of things that we will be looking for as we try to understand effectiveness. You know, to what extent does that correlate with increases in gene expression as the major driver for effectiveness? That may come from copy number variation. For instance, we know that hemoglobins have evolved in their role of modifying or controlling oxygen in nodules, in part through copy number variation. So that's one thing that we want to look at in pangenomes. Within a species, is there copy number variation for key genes like hemoglobin? And can we capture that copy number variation in breeding programs? We're also looking at that for carbon supply to the bacteria. We think we have dicarboxylate transporters now, and they appear to be part of a small multi-gene family that has been amplified in metacargo, for instance. And so copy number of key genes, like the genes that provide carbon to the bacteria may be important in optimizing nitrogen fixation.
Michael, I have a third question for you. It is a little bit technical. I hope I will be able to explain what I mean. You showed a very interesting graph. It regards the concept of effectiveness. You showed a graph in which you have the response, let's say the biomass production under that one on the left. You have the response of plant biomass under optimal and then under suboptimal reduced nitrogen. And effectiveness would be like a ratio, no? So yes. The genotype that does decrease the least will be more effective.
Yes.
Now, this concept has been used, I think, widely. It's been used widely, also in other contexts. For example, drought tolerance and water use efficiency, these things. Now, I've been using it myself pretty often, and I found sometime that this kind of ratio that you get is mainly influenced. For example, I will take drought tolerance as an example. There are genotypes that will have high intrinsic yielding ability, and then when water is missing, they will decrease very, very much, no? So that the ratio will be actually mainly a function of this yielding ability under under favorable conditions, no? And then when this happens, it turns out that since it reflects this kind of response under favorable conditions more than adaptation to unfavorable conditions, really, because of ratio construction. In the end, I mean, it may not be the most useful approach in identifying material that is well performing under highly limiting conditions. So I don't know if this is the case for also here, but then I was wondering what could be if there is something, I mean, possibly true in what I say, there would be, I mean, what is the potential value of different approaches? For example, like evolutionary approaches in which you are letting a population evolve under very limiting nitrogen availability so that you kind of shift this population, or anyway, you just indirectly select the material under limiting nitrogen conditions.
Yeah, well, I think ultimately that's what we hope to do is, through field trials, for instance, under low nitrogen conditions, look at the variation in breeding populations, whether those diversity panels and core collections that are feeding into breeding programs, or in nested association mapping populations, etc. So, we want to be able to test these various genotypes in nitrogen limiting conditions, although it's difficult to get completely nitrogen free conditions in these sorts of experiments, but we want to sort of try to improve our chances of success by finding low nitrogen conditions for these trials. So that's one thing. I want to come back to the point you made about whether the ratio is really the right thing, and we thought about that as well. I mean, it may be that you have an effective genotype which is 100% effective, but it's a slow grower, so it's able to get close to that optimal or maximal yield under mineral nitrogen simply because it's a slow grower. It doesn't have to fix that much. What I think you can see though in this left panel is that there's quite a bit of variation regardless of whether a plant is a slow grower or whether it's a fast grower. Even some of these fast growing plants have wide variation in nitrogen fixation effectiveness. So our hope is that we can combine effectiveness with yield so that we can get plants that are robust, but also are fixing effectively as well. And in practice, we won't carry out the experiments in the way we did here, where we were able to control. We had sort of plenty of seed, and we were able to grow the same genotype under mineral nitrogen versus nitrogen fixation. In practice, what we want to do is use 15N ratios, 15N to 14N ratios of individual plants as an indicator of nitrogen being fixed within that plant. So we won't have a separate plant as our control, if you like, our mineral control. We would estimate nitrogen fixation effectiveness using natural abundance of 15N and 14N. So that's our approach moving forward. And of course, we're aiming to get effectiveness combined with yield. So we're looking for plants that are robust as well as effective at nitrogen fixation. So really pushing this end of the spectrum, if you like.
It's clear. Now, is there any other question from? You can just you can raise your hand, or I think just talking. We are many. Well, then, Michael, thank you again for this very, very interesting speech, and good luck for your next few years of work in Australia.
Yeah, well, thanks, Paolo. It was a pleasure, and it's a bit difficult to have this conversation when we're sitting a world apart, and I can't see the audience. No doubt the audience would have kicked me to make me go faster because I used the whole hour in my talk, and I apologize for that, but I hope it was useful to some people.
Thanks. Goodbye,
Yeah.
Michael.
Thanks, Paolo. All the best.
Good day to everybody. Goodbye.